Greetings and welcome back. So, this is the third in a short series related to exploratory dynamics. And what we're going to do in this vignette is we're going to tackle the specifics of a case study in exploratory dynamics. You might call it the canonical case study. And it has to do with how it is that the chromosomes are captured by the microtubules, which eventually will pull them apart prior to cell division. So, just to reiterate, we've already made a point of chapter 4 of the Gerhardt and Kirschner book. And on the right, I am giving you my example of various case studies at different scales, different length scales, and different time scales. And as I say, we want to focus on the case of cell division. So, this is a classic movie, and for my purposes, all that I really want you to see is relatively qualitative. The DNA essentially is labeled in blue. That's fine, and there are these absolutely mesmerizing structures that are formed that are known as the spindle, and the green stuff that you see is tubulin, which polymerizes to form these so-called microtubules, and that is what affects the pulling apart of the chromosomes. And the thing that I want to say, and that maybe is not immediately evident upon looking at a video like that, is that what's going on beneath the surface is that these microtubules are growing and then shrinking and growing and shrinking and so on. And there's a molecular complex, which I will refer to as the kinetochore. You can see it here labeled in yellow on the left. And this is a point of attachment between a microtubule and the chromosome, and is itself its own interesting and beautiful and compelling story. How it is that by depolymerizing a microtubule could pull apart the chromosomes is a fun, interesting thing to think about. For my purposes, what I want to get across here is that we're going to invoke the mathematics that we wrote last time, which is the mathematics of the geometric distribution, in order to try and evaluate the time evolution of this particular exploratory process. So before moving on, I just want to persuade you that if you were to watch microtubules, what you would see is this so-called dynamic instability. I regret the absence of both spatial and temporal time scale bars, but suffice it to say, just take a look at the movie. What I'm hoping that you see is, you can pick a given microtubule, and what you'll see is that it grows and then it shrinks and so on. And the reason that this is so important is that that's the underlying dynamics that gives rise to the exploration, which I'm basically trying to point out. Now, one of the things that I guess I wanted to say, I've commented on it before, when we looked at the protein translation case, I always like to point out that famed movie where tRNAs are arriving at the ribosome repeatedly, it's misleading because most of the tRNAs that arrive will not be correct. Here, there's a similar point which has to do with errors and their correction. So just like in the case of translation of proteins, we want to make sure that the right tRNA is admitted into the ribosome for peptide bond formation. Here, we want to make sure, or I shouldn't say we, but the cell wants to make sure, there's a purpose, if you'll let me use that term, the cell wants to make sure that there are the exact correct number of attachments between microtubules and chromosomes. And I love to show this. This may seem like a non sequitur, but it's really not. This is an amazing, beautiful figure. I first learned about it when I was a professor at Brown. I used to go swimming every day with a guy named Ken Miller. He's a professor there, well known not only for his book on biology, his teaching of freshman biology, but also he served quite a bit as a witness in these cases in which there's some trial in which a teacher or a school system or something is challenged for teaching evolution. And in swimming with him, we would do laps together. He once said, you should come take my freshman biology class. And so I basically signed up, not literally, but went every day to a big giant lecture hall across campus from the Barris and Holly building where I was, and listened to him talk through biology. And one of the things that he explained was this particular point, which is the fact that out of all the primates, humans are the only one with 23 pairs of chromosomes. All the other species have 24. And his point was that that leads to a prediction. So this is a tangent, just for a moment. The tangent is this. So I wanted to show this because I want you to know there are 23 pairs of chromosomes. It means there need to be 46 attachments. But the reason for the little tangent is to say that there's a prediction of this idea that we have 23 and other primates have 24, which is that one of our chromosomes represents a merger. And what Ken told us about is that there should be telomeres on the interior of this merged chromosome. And I leave it to you to go do a little bit of detective work. But it's really super interesting that such a thing exists. I also wanted to mention this quite cool paper that Ariel Amir and Dan Needleman were responsible for. The two lead authors are both former Caltech undergrads, Gloria Ha and Paul Dieterle, people that I admire a great deal. And I'm not asking you to look at this in any technical detail. All I want you to note is that in part D and in part F, in part D, they're showing the mismatch between how many times there's an error in the number of connections made to chromosomes. What do I mean by that? There need to be 46, not 45, not 47. There need to be 46. And then the cell is licensed to divide. And what they did, is shown in E, is they caused the cells to divide prematurely, and then they counted the number of errors, if you like. So you can see in part C up here, it says delta n is generally 0, which means that the number of kinetochores, or the number of connections, is 46. And on the bottom, you can see that if you cause a premature cell division, then there will not be a correct 46 capturings. Why is this important? Because I'm going to make the claim that this exploratory dynamics, which is the heart of what we're talking about, is predicated or is the basis of this kind of error correction. So what I'm going to do now is relatively qualitative, and what I mean by that is I'm going to show you how, using the mathematics of the geometric distribution, which we did last time, we can set up a toy model of the search and capture process for chromosomes. I basically have two different versions of a model. The lower left is a one-dimensional search where we presume that the microtubule is going in the correct direction right from the get-go, and the part C, the three-dimensional search, imagines that every microtubule can grow anywhere on the unit sphere, and so that means most of the time it will be growing in the wrong direction, and therefore is certain to miss the chromosome, and it's only over a little cone of possible directions. So we'll see how to write down the probabilities. So what I'm showing you here is, in a certain sense, a homework for people to contemplate. And what it's doing is it's organizing our trajectories by number of failures. So in the top left, that particular microtubule grew. It grew to the left, and it hit the chromosome, and so it was a success right from the get-go. In case I equals 1, so that's the second one, what I want you to notice is that this guy grew, it didn't quite make it to the chromosome, it shrank, and then it tried again, and it succeeded on the second try. As you can see, the structure of these probabilistic trajectories are all of the form 1 minus ps to a power. The power is the number of failures, followed by a ps, because remember, ps is the probability of success. Just as we did in the previous vignette, I can do the mathematics of this geometric distribution. We already know how this plays out because we worked out the time, the average time, or the average number of steps, the average number of failures. And you can see that we get this quite cool equation. Now here, what I'm showing you, this is, in a certain sense, also a homework, meaning anybody that wants to seriously engage with this needs to sit down and do the math for themselves. But here's what I would say. There's kind of two probabilistic things going on. First, does the microtubule grow in the right direction? So FR is the probability of when you flip a coin. This is an unfair coin. The first coin flip is which direction do I go in? So it's an unfair coin. If I get the small value, then I'm going to go in the right direction. If I get the large value, I'm saying I've divided 0 to 1 into an interval. There's a very small slice of 0 to 1, which corresponds to growing in the right direction. And so I flip this unfair coin a bunch of times, and probability of going in the right direction is what I'm calling FR. And then what we do is we basically acknowledge that I can grow in the right direction and still not make the chromosome, or I can go in the wrong direction and certainly not make the chromosome. So the binomial distribution tells me about the sum of all of those different trajectories. And one can work through the details of this. And what you find is that the search time, as a function of the catastrophe rate is the rate at which the microtubules depolymerize, that there's a minimum in the search time. I'm not really trying to make a big deal about this minimum. It's just more to show you the philosophy of how to think about one of these exploratory problems. I think the last thing I'm going to say, and I'm not going to go further, although there's a whole story that has to do with the power. I just want to note that in the early 1970s, John Hopfield and Mark Hirschner were both at Princeton, and they were both thinking about what amounted in the end to the same thing. And what I mean by that is that both of them noticed an apparently futile hydrolysis of GTP. In the case of Hopfield, it was in the context of protein translation. In the case of Kirshner, it was in the context of microtubules and their growth. And I just think it's super interesting that, in a word, exploratory dynamics costs. In other words, for the system to do this, we've already been through this analogy with building a train between New York and Boston. It costs. And so it's very interesting to evaluate what the cost is in ATP equivalent units, but also to try to understand what the benefits are. So this is, for the time being, going to be the end of what I wanted to say about the exploratory dynamics, but I want to leave you with this observation. And the observation, I guess, is that we've had 350 years of this glorious update rule, calculus-based dynamical systems, however you want to label it. I'm being a little sloppy in my language, but I know what I mean. We've used these update rules. We say thing now is thing before plus some sort of a update, and the update comes from F equals ma, or the law of mass action, or from the heat equation, or whatever. The assertion that I was making, and I think it's, in some sense quite revolutionary on the part of Gerhardt and Kirshner, is that no, there's a different kind of dynamics which appears to be unique and necessary in the setting of living organisms, which has to do with exploration. And the reasoning here for me is that the system cares about the final state. It needs to guarantee a final state, regardless of what the initial condition is. People have asked me when I've talked about this, they say, well, that's not quite right, because if you have a bowl and I release a marble from anywhere on the bowl, it'll always go to the bottom. But that's not really what's going on here. And the reason I say that is there's no bottom, there's no bowl. In other words, I can state it differently. I could do an in vitro experiment in which I take an optical trap and move these microtubules, move either the source of the microtubules, move the chromosomes. It's still going to work. The exploratory system will continue to find its terminal privileged state, and to do so will have to have consumed energy. I hope you found that interesting. I suggest that you go to Libris and buy a copy of Gerhardt and Kirshner's book and check out Chapter 4.